right, we are back with part 2. We are on Model 2, reproductive isolation. First, let's go over the answers to this second part of part 1. So I'm on page 4. I just want to make sure you guys have the right answers to that. This is good practice. You're going to have to do this on quizzes and tests. Okay, so just saying, giving you a heads up. This is good practice for you. So it's number 8, and you're describing what the population is, what type of selection they're in. So for 8A, we have finches with a small beak that cannot crack open seeds. So a population in which finches with small beaks cannot get food. Okay, that would be directional selection. B, human babies with high or very low birth weights have a higher mortality rate. So we have very high birth weight, and low birth weight are bad. So we have selection against that, which means we're selecting towards the norm, towards the medium phenotype, which would be stabilizing selection. And then you have number 8, part C, a population of seed-cracking finches feeds on seeds available in 2 sizes. So you have either small seeds or large seeds. So in this case, the smaller birds would be able to eat the small seeds easier, and the larger birds would be able to crack open the seeds for the large seeds easier. In this case, it would be disruptive selection. And D, overfishing in 2 rivers in British Columbia, Canada, where larger salmon are preferentially caught. That would be directional selection, because you're selecting for that higher weight of salmon is going to be caught. So over time, that would lead to, in the population, on average, they would be smaller in mass. All right, so we're talking about now how you actually get 2 species from a population, and what actually makes a species a species. So the big thing is reproductive isolation, and that's what we're going to go over here. So what makes our species, what makes any particular species a type of species? How do we define that? That's what we're looking into. It's all about who can reproduce and who can't, okay? Or who can reproduce fertile offspring, fertile and viable. They have to be able to survive to reproduction as well. So we have different types of reproductive isolation, and let's just go over each of these types on this page. First of all, we only have one scenario in which these individuals are the same species. So you have A and B, type A and B. You have mating results in viable, fertile offspring. All viable means is that they're able to survive to reproduction. So they're strong and viable, able to survive. Fertile means that they can reproduce. So when A and B, when they reproduce, mating results in viable, fertile offspring. So for those organisms, A and B would be the same species. I put a little check mark by them. Okay. Organisms C and D, mating results in viable but infertile offspring. So it means those organisms can survive to reproductive age, but they are infertile, meaning they're born sterile. In that case, that would not lead to something that is the same species. Think of like a mule scenario, in which I actually gave the answer to one of these on the back. But if you have a donkey and a horse mating, they make a mule, which mules are great. They're very good for doing work around the farm, but they're sterile. They can't reproduce. And that would make horses and donkeys not the same species, because their offspring cannot reproduce. So they're not considered the same species. Mating results in, for E and F, mating results in weak offspring that does not live to reproduce. So these offspring would be inviable. They're not viable. They're not surviving a reproduction, in which case they cannot be considered the same species. Because if they're too weak to survive to reproduction, then evolutionarily it doesn't matter. They can't pass down their genes, so they can't be the same species. G and H. G and H, organisms live in separate geological areas. Okay, so they do not live in the same area at all. Mating is not possible, so they are not the same species. I and J organisms do not have compatible reproductive organs, meaning part A does not fit into slot B. Part A does not fit into slot C, therefore they cannot reproduce because the parts don't fit together right. K and L organisms feed from different sources, meaning they're unlikely to encounter each other in the wild. If they're unlikely to encounter, then they are unlikely to mate, meaning they can't be the same species if they never encounter each other. Okay, so you'll notice that some of these have to do with space. When they're actually, are they actually probability-wise running into each other? Are they able to even encounter each other in the wild? And some of it has to do with what we call post-zygotic isolation. Let's explain these concepts fully. Okay, so I wrote here, I categorized these top parts versus these bottom ones into two different ones. And I'm not including A and B in this because A and B are the same species, so they're not counting in this. So for C, D, E, F, they are in what's called post-zygotic isolation. Zygote just means a sperm from male and then egg from a female come together and they form a zygote. So when fertilization occurs, that's a zygote. So that's post-zygotic isolation, meaning they were able to form an offspring, but it was after that zygote was made that made them reproductively isolated because they either didn't survive reproduction or they were infertile to begin with. So it's post-zygotic isolation that kept these guys from being the same species. On the bottom here, we have GH, IJ, KL. They are in pre-zygotic isolation, and there are different types of pre-zygotic and post-zygotic isolation. There are different types. But each of these are pre-zygotic isolation because they're not even getting the opportunity to mate. So they're not creating a zygote, which makes it pre-zygotic isolation. So G and H, they're geographically separated, no way of encountering each other, pre-zygotic. I and J, part A does not fit into slot B, so because the parts don't fit, that's pre-zygotic isolation. And then you have K and L, where they are, whether it has to do with when they're feeding, like maybe one feeds at night, one feeds during the day, but they might live in the same area, but they're just not encountering each other. So they're not likely to mate at all. So those are all examples of pre-zygotic isolation. They don't even have a chance to form a zygote at all. They don't form offspring at all. Okay, so looking on the back side here, the ones that are able to reproduce offspring are A and B because they are the same species. And then C and D, E and F, these guys, C and D and E and F, those are in post-zygotic isolation, so they can produce offspring, but only A and B are that same species like we talked about. Okay, so A and B would only be the only ones in that scenario, which would be the same species. Okay, if you haven't done this already, again, go through all of these numbers. I'm just going to make sure you have the right answers for number 12 through 16. Okay, so 12 through 16, 12 would be no. That's an example of post-zygotic isolation because they're making a sterile offspring. 13, no, it's pre-zygotic. It is what we call behavioral isolation, and we'll talk more about each of these different types. This is just an introduction to it, so we will get more into this in another video later. But the answer would be no, pre-zygotic, it's behavioral isolation. 14, no, entire population is changing frequencies in the same direction. So 14 through 16 discuss, can these different types of selection lead to speciation? Okay, so if you have directional selection, they're moving in one direction. Okay, so again, back to this, moving in one direction. So for example, here, selecting for a higher birth weight. Since the whole population is moving in one direction, that would not be able to make a new species because the whole population is changing. You're not having separation. 15 would be yes, because allele frequencies are changing in opposite directions for disruptive selection. So over a long period of time, you could have, for example, these two, we talked about these two different mice, for example. Let's call them mice. So you could have mice that have, if they're really small, it's good they fit in those small spaces, or if they're really large, they get to maybe eat a specific food source that normally the small ones or medium ones would not be able to get to. Over a long period of time, this could lead to reproductive isolation because you could eventually have these guys reach a point, reach a point where they're either behaviorally they're not in the same niche at all. They're not even occupying the same space. It could be that eventually you don't have slot A fitting into slot B, so they can't reproduce. It could be a number of things. Like this could eventually lead to reproductive isolation, which could lead to a different species over time. And then 16, would stabilizing lead to the creation of a new species? That would be no, because they're all converging to the same phenotype, similar to directional selection. The whole population is changing, so you're not getting the separation of traits. You're not getting a split. Anytime you get a split, you could eventually reach reproductive isolation, which could lead to new species, and that is how new species occur over time. All right, so that is our introduction to speciation, and we're going to have another video on this. We're going to do our part C, genetic drift, next. And we will, I did have a fun activity I wanted you to do for genetic drift, but unfortunately it's definitely an in-person thing, got to be there the whole school. You may have seen this last year. I had a bunch of kids coming around having you taste paper. That was for the genetic drift activity. But sorry guys, kind of stuck. I might be able to find another activity that helps demonstrate that. So we may go into genetic drift further past this. We will. But this is our introduction to it. So you get genetic drift if you have, I call this guy also effective small population size. Because you get genetic drift very easily in small populations, whereas this wouldn't occur in large populations. So first I had you go through each of these generations and figure out how many of each there were. At the very beginning, you had 4 of each color. Okay? And then it says one of these, and they're even. Okay, so 18 asks about it is even. Okay, and how'd you feel out all these? But in 19, it says that you had a death of an individual with 2 light alleles. If you take one individual away, that gets rid of 2 of the light alleles. So here's what you're left with, second generation, because that one individual died. You're having less light alleles, which continues to the 3rd generation and 4th generation. And it was all because of that one individual dying. So in genetic drift, it's shifting to medium and dark phenotypes and genotypes. And 20 asks, why is this occurring? Why do you think this is occurring? And this is largely due to chance. Because if you have one individual dying in a small population, that can have a dramatic effect on the allele frequencies. Because you take one individual away, you're taking away 2 alleles, which means that for the rest of the population, that just takes away, if they're even at the beginning, then that automatically leads to a skew in one direction. So just due to chance, now you have more of the medium and dark ones mating. Over time, you're going to have more medium and darks and less light. And that was just because one died due to chance, just happened to die. And again, you guys know you can die in any kind of way. Any kind of way, just say it was a random, got hit by a car. Okay, well, you get hit by a car, that individual is now out of the population. And now it skews the whole small population because there were not that many individuals to begin with. So you take all that individual's genetic information out of the population. And 21 asks, what would happen for the light allele to occur? Now, you could have that pop back into the population. Say if you had two heterozygous individuals, create a lighter, an individual with two light alleles, because you do have some intermediate phenotypes here. So if they were to be the ones to mate, you could have that pop back into the population. Or if migration, you could always have another one from a different population, migrate to this population. That's an option. Or mutation, but those first two could be unlikely. Especially mutation would be very unlikely. And 22 is kind of like the main point of it. 22, if this was hundreds, we gave you, instead of these tiny little squares, I gave you one that was the size of this page, and it had a ton of dots that you had to count. And you had the death of one individual. Would the death of one individual have a dramatic effect if this population was 10 times as large, maybe even 100 times as large? The answer would be no, because you're not skewing in one whole direction. Population is large. So this is what we call genetic drift. It's just the effect of small population size, meaning that if you have a chance event, it has a more dramatic effect if it's a small population rather than a large population. All right, that is going to conclude today's. So we had just part 1 and 2, as well as that introduction, which goes over kind of what to expect, as well as you get to see Jack in that video. So go ahead and view that. You should have just a reminder, you should have your active reading guides due today. So you submit it to that assignment. There is an assignment on Google Classroom. You'll just have to scroll down a little bit to see it. You're not turning in the POGL because you kind of needed it for this lesson. So guys, you just I have to trust you more in these online lessons, honestly. I don't get to see what you guys are doing. You could skip it, but other than you're missing the entire lesson. And we will have quizzes on Fridays. I think that's kind of they'll be open note, obviously, because I know you will use your notes. I'm not oblivious to that. But yes, you will have open note quizzes, and I will quiz you over this stuff, but you're welcome to use any notes. What else did I want to talk about? Oh, tomorrow we are going to I want to go into speciation, a couple of these concepts more, but we're going to spend a little time talking about how natural selection has an effect on viruses, just because that's so topical right now. So we are going to do an Ebola disease detectives activity. I just think you guys will find this interesting, and it's very relevant to what could have happened in the emergence of coronavirus, honestly. This is how viruses undergo natural selection as well. And this kind of goes over how it applies to viruses, because they need a cell to replicate, they need a cell to reproduce, which makes them unique organisms compared to other things. So that is what we're looking at tomorrow. So tomorrow will be all about this Ebola thing, which I did not print out for you, but I will give you the link to this, and you can just you can take notes in a notebook, paper. Yeah, I would do that for the open note quiz. So you're not going to turn this in. But for the open note quiz, you're going to want to have this down. And you can print it out, or you don't have to. You can just put it in notebook paper. You shouldn't have to print it out if you don't have a printer. Let's see. I think that is it. I'm also posting a video. You should, over natural selection, one that I find that's good. Make sure you watch that. It's a good one. And March Mammal Madness, that will be posted as well. So make sure you are keeping up with us, okay? If you wanted to start your chapter 24 active reading guide, you are welcome to start that. That will likely be due, I would say next Wednesday or next Monday. Probably next Wednesday. Give you some time to do that. And then we'd have just one more POGL before we will have a test of some sort in some way. All right. You guys have questions, you have concerns, feel free to email me. If you want me to If you want me to include something fun in here, let me know what you would like me to include. I may listen to you, I may not, but I want to make this as interactive as possible, even though it's just me talking to myself in a room. All right. You guys have a great day. Take care of yourselves. Stay active. Make a schedule for yourself. Make sure you're maintaining social distancing, because the longer more people ignore this, the longer we're going to have to do it. And I'm going crazy. I don't know about you guys. So, all right. Salute. Bye-bye.